All right, welcome to topic 8 in marine aquatic ecology. We continue within the population unit of this course, and our subjects today, this week, are methods for estimating the absolute size of populations, methods and important reasons for understanding fluctuations in population size, and what those fluctuations mean for fitness of organisms. And then we begin the topic of interaction between two populations, types of interactions that we see in nature, and start in on competition in more detail as an interaction that reduces the fitness of both competitors. And we use Lotka-Volterra theory for understanding these interactions. Okay, so today we continue at the population level of an organization, and we want to quantify the ideas described verbally in Darwin's chapter 3 of The Origin of Species, the struggle for existence. And the ideas are populations increase exponentially until resources become scarce. And there's a struggle, a competitive struggle for those scarce resources. And we already have equations for that exponential increase and for the reduction in the per capita rate of increase, which is little r, as the population density increases due to the fact that as n increases, the amount of resource per individual decreases. If the amount of resource per individual is proportional to the per capita rate of increase, then the per capita rate of increase will fall linearly as n increases. So the per capita rate of increase is a measure of the relative mean fitness of the population. So this basic relationship, a decrease in R, which is fitness, as N, which is density, increases, this shows how levels of biological organization interact. And the interacting levels are organisms and populations. So fitness, or little r, is a property of individuals, but we can define it in terms of the property of the population, the r for the population. So that's the mean fitness of individuals. So if we compare two types of individuals with two different r values, then we can predict that the individual with a higher r value will increase its population of descendants faster in the struggle for existence. Okay, so fitness, or little r, determines the rate of increase of the population of descendants, and population density, which is n, determines the mean relative fitness of individuals in the population. Okay, so we also know from previous lectures that the pattern of dispersion, which is a property of the population, will influence fitness of individuals. For example, individuals disperse according to the ideal free distribution or the despotic distribution to maximize their fitness, which is a property of organisms. So properties of individuals, individual organisms influence properties of the population and vice versa. So we now discuss in the simplest terms how ecologists estimate the absolute population density of a given species within some fixed area, which is a common problem in any field study. You want to know how many individuals are there, the absolute number. Having a good estimate of population density is valuable for both a basic understanding and for practical economic and conservation concerns. For example, regulation of the amount of fish harvest and setting up a plan for sustainable harvest, for control of an invasive species, for conservation and recovery of a threatened or endangered species. Definitely you want to know how many individuals of an endangered species are present in different areas. And so note here, though, we're not talking about relative abundance, although that's also important and useful to know. Relative abundance, so in other words, the abundance relative to some other location or some other time, is estimated easily just by taking a random statistical sample using the same methods at different times and places. So this is a simple problem. A more difficult problem that we discuss now is estimating not relative, but the real total abundance or density of individuals of a particular species at some place and time. So estimating the absolute number is done not by counting every single individual, unless it's possible to do so. Sometimes it is if it's a rare species. There are not very many to count. But rather, what's used is some variation on the basic method called mark and recapture. So basically, you mark some random sample of individuals, let them back into the environment, then capture a random sample of individuals, and note how many were previously marked in your sample of recaptures. So individuals can be marked in a variety of ways depending on the species. Fish and larger invertebrates with hard body parts can be marked with plastic or metal tags on their fins. Large whales can be identified often by natural markings or patterns of barnacles on their skin. Even small whales, dolphins. Many mollusks with shells can be marked with colored nail polish, waterproof nail polish. Of course, birds can be marked with tags attached to their ankles. So there are many methods to do this. All right, here's how the theory works. Let's say there are 100 individuals in a population. So this is the number we want to estimate. We don't know what it is, but let's just assume that it's 100. So n is equal to 100. So how do we estimate this number if we can only see and we can only capture some small subset of what's there? How can we possibly estimate the total number? Well, we first capture a subset, a random subset within some defined spatial area, a random subset, say 20 individuals, and then we mark all 20. Okay, let's call M the number marked. So M is equal to 20. So now the proportion marked is P equals 20 over 100 is 0.2. That's M over N. Okay, the number marked in our sample divided by the total N that we want to estimate. We don't know what it is yet. So we can also think of P as a probability that a random individual in the population is marked. Now we release those M equals 20 individuals back into the population and assume they behave as they normally would, move around normally, and carry out their normal functions. All right, then at some later time, we go back and capture another random sample of individuals in the same area. Let's say we capture 50 individuals. Let's call this number N sub S. Then for those N sub S 50 individuals, we count up the number marked in our new sample. Let's call that number M sub S. What do we expect this number to be? Well, if we sample at random, then we expect that a proportion P in our sample should be marked. That's the probability that any individual is marked. So in a random sample, a proportion P should be marked. So M sub S, we expect to equal 50 times P, which is 50 times M over N. Which is the number of our sample, number in our sample, NS, times the number marked divided by the total N. Okay, we don't know what the total N is, but we do know the number in our sample, and we do know the number that we marked. So now we just solve for N, what we want to know, using a little algebra. We solve to N to get our estimate of the total absolute number of individuals in the population inhabiting the area that we randomly sampled. We solve for N to get our estimate of the total absolute number of individuals in the population inhabiting the area that we randomly sampled. So that estimate is, we just do the algebra. If we solve for n here, that n is equal to number in the sample times the number that we marked divided by the number marked in our sample. Okay, so in other words, we've got the individuals that we're trying to count, and we have the sample. And the proportion marked in the sample should be the same proportion marked in the total. So in other words, the number marked in our sample is to the number marked total as the number in our sample is to the total number if everything is sampled at random. So the sample is a microcosm of the entire population if we sample at random. So the proportion marked in the sample is assumed to be the same as the proportion marked in the entire population. Okay, so we just form this ratio equal to that ratio, and therefore this is our estimate right here of the total population in the area that we randomly sampled. So that's basically it. That's a basic idea in mark recapture studies. They're very, very easy to do in invertebrates like snails and crustaceans that are easy to mark. So mark recapture studies to estimate absolute n, of course, have assumptions that may or may not be met in nature. If they are not met, then adjustments and corrections need to be made to the estimate. And much of the literature is all about those adjustments and corrections, and they can get fairly complicated. The main assumption is that the probability of being caught is independent of the probability of being marked, which implies that this probability is the same in both marked and unmarked individuals. For this probability to be the same, the probability of dying or migrating away must be the same for marked and unmarked. That's the basic logic of mark recapture studies. So if you can assume that the mark does not affect behavior in a way that interferes with your sampling, then you're okay. Sometimes it's not okay to make that assumption, but sometimes it is. Here's an example of a marine mark recapture study. So Cancer pagurus, the edible crab in the UK, this is an example of estimation of the total population density on an offshore bank off the English East Coast. Bell and colleagues in the UK did this in 2003. These crabs were marked with just indelible waterproof paint. So crabs were caught during continuous trapping field trips using crab traps just to monitor catch statistics, and they were painted. So then the trapping continued over several weeks, and the researchers noticed if they re-caught marked crabs and how many. So they've got observed recaptures. This is the mark recapture data on different times. And these are the marked ones that were never recaptured. These were the recaptured marked ones and the marked that were never recaptured. So then they showed box plots of their estimate of n, population size, in several experiments here, with X marking the mean of the estimate within the box quantile. So this is easy to do. So the result is that the estimate of the absolute number of crabs on this particular offshore bank was somewhere around 5,000, pretty consistent in several trapping surveys. So this number of around 5,000 or so can then be used to calculate the total annual productivity of the crabs and to regulate the total number of crabs taken by the fishery to ensure sustainable harvest. In order to regulate the total number taken, you really have to know the total number are there, which is the reason this is very important to be able to do these mark recapture studies. So that's the basic idea how absolute n is estimated in nature in a field study. So that's basically done all the time. But in addition, and for other types of questions, um, however, we just want to know the relative n, how it changes from time to time or is different from one place to the next, from one habitat to the next. How much it changes over the seasons and years, and there's no urgent need to know the absolute number at any time. So there's many examples of monitoring populations at a particular location, meaning watching the relative numbers in a uniform sample taken the same way every time, watching those numbers fluctuate. And in these types of studies, we don't know the absolute number. You just want to observe and explain why they fluctuate. So let's consider that question. Population fluctuation, changes in the population density at a particular location from time to time, why those changes occur, how we can quantify those changes, and explain why they are occurring. For example, take a constant sample of water from some location in the ocean and under a microscope count the number of phytoplankton cells, for example, surf diatoms. Phytoplankton, they're common during surf conditions on the beach. So Chaetoceros and Asterianella, for example, count those numbers in the microscope and keep track of the fluctuations in those sample numbers over time. And so if you can assume that your sample is random and you make the same effort every time, then you can conclude that the real absolute population fluctuation is mirrored in your sample, with some uncertainty. So here we're not attempting to estimate the absolute number of diatom cells in the water at any particular location. We're just looking at the fluctuations in our sample, which is easy to take. No mark recapture is necessary. We see some very pronounced seasonal patterns with low numbers in the summer, high numbers in between in the winter in both species. That needs some explanation. Another example, population fluctuation in lemon sharks in a nursery area in Bimini. In this case, sharks were caught with a gill net, in other words, sampled, and the number caught per unit effort for the number of nets and the amount of time, that catch per unit effort, number of individuals, was plotted each year under the assumption that the catch per unit effort is a proxy variable for the total population size. So again, here the authors weren't interested in the absolute population, just the fluctuations over time, which they estimated with a sample. So catch per unit effort, or CPUE, is usually assumed to be adequate to estimate population fluctuation. Here's another example of trawl fishing results, number of fish caught or sampled in a trawl net. Again, not the absolute numbers, just the relative numbers. And just by looking at relative numbers, you can see patterns. Okay, so we've got haddock here. These numbers in the red obviously are fluctuating from one year to the next for whatever reason. But the skate and the flatfish numbers are also fluctuating, but in a sort of a mirror image of the haddock to some extent. So as the haddock decreases here from 1980 to 2000, the others increase. So is this because they're competing for the same invertebrate echinoderms as a food source on the sea bottom? Interesting pattern, isn't it? Right, what causes population fluctuation? Well, it's possible there are internal factors intrinsic to the population. For example, the age structure is not at equilibrium. There's also time lags in response to the environment, and there can be random variation in births and deaths. And these are important, but not as interesting in many cases, a lot of the time, in as another reason, which is that the mean fitness of individuals in the population fluctuates, often in response to a varying environment. And it's important to know what that variation is for conservation, management, and just for basic understanding of the ecology of the species. So that fluctuation then can be a clue to the factors that limit fitness of individuals and provides interesting information about the important ecological relationships of individuals regarding their survival and reproduction. So, understanding population fluctuations is often a key to understanding the relationship between individuals and their environment and factors determining their success in survival and reproduction. Okay, so let's look at it in more detail. One method of determining what environmental factor is most important in controlling population size and growth is what's called key factor analysis or K factor analysis. So this is a method that simply finds correlations between population density over time and some key factor that is hypothesized to control it. So this method is similar to sensitivity analysis, which is related. Both want to understand factors that correlate with population density over time or space, but sensitivity analysis is a method that finds which life stage population density is most correlated with total population density. So the life stage that is most correlated is then inferred to be the most sensitive life stage of the species, the life stage whose success or failure is most important in determining the total population and health of the population. So this is the life stage most important for conservation of the population. All right, so that's different from key factor analysis, which does not necessarily look at the life table, the life stages. It looks at external factors varying and what the most important external factor is, and that is the key factor. So key factor analysis basically looks at the correlation between lambda on the one hand and each of a suite of survivorship values within the life table on the other hand. It's usually done on the log scale. So we've got a bunch of lambdas over time, and these lambdas are the sums of the survivorship and birth rate, the net reproductive rate in the life table over several time intervals. And this total survivorship from one time to another time, this L here might be the product of several survivorships within that year or time interval. So survivorship month 1, month 2, month 3, month 4, month 5, and so on, across the seasons or whatever, might be the product of several of these, before you multiply by B. Right, so this is usually done on the log scale because a product on the raw scale is is the same as the sum on the log scale, and so the effective lambda from one generation to the next equal to n2 over n1, not the equilibrium lambda calculated from the life table, but just the effective lambda from one generation to the next, is equal to the product of many survival components and the birth rate. This lambda is equal to the product of many survival components. These might be survival of different months or survival to different types of processes happening in the environment. And they're all multiplied on the raw scale, but you sum them up on the log scale. So you can take the log of lambda, and that log of lambda is equal to the sum of the logs. Okay, so key factor analysis can be easily visualized on a plot like this, with time on the x-axis and the log of the components of lambda on the y-axis. So here's the log of the first component of survivorship. Here's the log of the second component, and so on. Log of the third component. You can keep drawing these graphs of all of the components of lambda. So you can just visually inspect this plot and see which component of lambda is the most highly correlated with the total lambda. It looks like S sub A, whatever component that is highly correlated with the total, which indicates that the fluctuation in lambda from one year to the next seems to be caused primarily by fluctuation in this particular component, which is the key factor. Okay, why the log scale? Because you can just sum up on the log scale. So the total here is equal to the sum of all of these subset graphs. Right, you can also do a scatter plot of the log of lambda against the log of the component. Usually a survivorship or count of individuals is a certain age, and do a linear regression or just a correlation test in R. And so the plot that has the highest correlation would then be the key factor. So a nice marine example of a key factor analysis is the one of Bailey and colleagues. This is in Seattle, Washington, in 1996 on the fish pollock, in which they plotted the log of abundance over time for each of several ages or life stages of the fish in several years, okay, in samples. And this shows survivorship from one stage to the next is basically linear on the log scale. So this is basically a type 2 survivorship curve. Remember, there's type 1 would be the K selected, would be basically flat at first and then come down towards later. And an R selected would be low at first and then sort of level off later. So this looks like roughly a type 2 mid-range between an R and K selected species. All right, so then they wanted to know which life stage mortality rate is most correlated with the total generational mortality rate, which is the product of all the stage mortalities. The product of all of these stage mortalities. And that product on the log scale is the sum of the logs. So their table 2 shows all the mortality values, or K values. This is the generational K. It's the log of, basically log of lambda. And it shows each of these components that contribute to that. And the table 3 shows the correlation coefficients between each stage mortality and the overall generational mortality, with the sample size n being the number of years they have observations for. So the general conclusion is the most important sources of mortality in this fish species during these years were larval and juvenile mortality. So both of these were excellent predictors of overall mortality across one generation. Okay, this is the overall mortality, generational mortality here. And the larval mortality is this right here. And then the juvenile mortality are these bars right here. See the larval and juvenile mortality are pretty well correlated with the total overall mortality. So therefore, management and conservation of this fish species would be most effective if it concentrated on the larval and juvenile stages. So transition from larval to juvenile is the stage at which this fish starts to school and attract predators, which can inflict substantial mortality, which makes sense. Larvae also feed on plankton in the euphotic zone. So this result suggests two hypotheses that can be further tested. Perhaps first, annual variation in predation rates on juveniles is responsible for most of the annual variation in the pollock population mortality. Or second, annual variation in phytoplankton densities is responsible for most of the annual variation in the pollock population. So the food of the larvae or predation on the juveniles. So a bit of further research can perhaps focus on testing these hypotheses. All right, so we want to know what the most important factor is that's responsible for variation in fitness over time, which is population fluctuation. We break down lambda into its components and watch those for several generations or years. And then we calculate the correlation with overall lambda and its components. Important to distinguish this from sensitivity analysis. Okay, so this is also related to elasticity analysis, just proportional versus raw scale. And it's similar to key factor, K factor, critical factor analysis, but there's no field data involved. And the goal is once you have a life table, then it's purely a mathematical question. You want to know how sensitive the life table lambda is to small variation in each of the components of the life table, each of the numbers in the life table. Elasticity is the slope of the relationship between the life table component and the life table lambda divided by lambda. In other words, it's the proportional effect. Sensitivity is the elasticity is the proportional effect. Sensitivity is the raw effect of the same component on the overall lambda. So a nice example of sensitivity or elasticity analysis is Heppel's colleagues, Selena Heppel in 96, on the endangered Kemp's Ridley sea turtles. So the question here is, what stage or age of the sea turtle should recovery efforts focus on? Because this is an endangered turtle and it needs to be recovered. So should efforts focus on the first year of life or on later stages, on subadults or on adults? The main threat for the sea turtle is bycatch in fishing nets. And the main conservation interventions have been beach protection during egg laying and hatching from 1978 on. And then in addition, laws requiring an escape hatch in trawl nets. That's the turtle excluder device, a nice little invention in trawl nets that allow these turtles to escape from the nets so they don't drown. So should there be additional protections and what should they be? People have proposed a Head Start program in which conservation groups collect eggs that have been laid in beaches and then bring them into a facility where they're hatched and fed for a year or so and cared for very well, then released back into the water. These Head Start programs are very popular because baby turtles are cute. And it's also fun to watch them hatch out of the eggs and take care of them. But Head Start program is expensive, so it's useful to know how effective it is. if Head Start increases the survivorship of hatchlings, what effect will that increase have on the population lambda, the overall lambda calculated from the life table? So this is a sensitivity or elasticity analysis. Once we have a reasonable life table, then we can make small changes in the numbers in the life table. Small changes in the age-specific survivorships and fertilities, and then calculate the lambda after you make those changes and see what effect it had. So independently of the rest of the life table. So we can increase age 1 survivorship as expected from a Head Start program, and then see how much effect it has on the population lambda. If it has a large effect, then that would be an argument in favor of spending the money on a Head Start program. So the sensitivity analysis was done by Heppel and colleagues in '96. It was found that increasing age 1 survivorship has only a very small effect on lambda. So this survivorship table shows you the effects of the different management actions have had on the population. So you have a model that assumes they reach maturity in 8 years. So these are the survivorships for pre-1978 when there was no management. And these are the survivorships 78 to 90 when the beach was protected. And then post-90, the beach protection plus the turtle excluder device regulations, which reduced incidental drowning in trawls. So you can see that the survivorships are going up from the previous time when there was no management. Okay, so here the beach protection increases survivorship of eggs and hatchlings, doubled it. In post-1990, the turtle excluder device increases survivorship of subadults and adults. All right, so the overall growth rate went from a decreasing population, lambda is less than 1, to a slowly increasing population. Okay, and same thing on these different models. If you assume maturity at 12 years or maturity at 16 years, these management interventions have had a positive effect on the population lambda that you can calculate from the from the life table. Right, in the sensitivity analysis, they found that increasing the survivorship of subadults has the greatest effect on lambda. So that's the elasticity or the sensitivity. So that these bars, no matter which of the models you had, the subadults, increasing the subadult survivorship increased the overall population lambda by the largest amount. And so, the hatching effective increase of the hatching survivorship during the Head Start program on the overall lambda had one of the smallest effects, and fecundity didn't have a large effect either. All right, so the Head Start program is going to have probably, look at this is 0.1 versus 0.5. So the Head Start program is going to have like 1/5 the effectiveness as a program that focuses on the subadults. So this indicates that the Head Start program will probably not be a good way of investing money in this turtle species conservation recovery. Unfortunately, rather investing conservation money on improving survivorship of subadults would reap the most benefits, which is a much harder proposition since subadults are scattered throughout the ocean. Although they certainly do benefit from the turtle excluder device. So after this paper was published, the Head Start program in Galveston, Texas was abandoned after 15 years of operation, funded mainly by donations, mainly by school children who heard about it in their classes. So the donations continue, but they are used now for more research. Beach closures and the trap door requirement in trawl nets are still in place. Biological benefits of head starting programs may be overestimated for turtles. And we need to look at the life table, do some more sensitivity analyses. And maybe that will guide us toward more effective management strategies. Okay, continuing with population fluctuation and its causes, we're going to look at internal causes of fluctuation in population. So if the age distribution changes, then the population size will fluctuate simply due to that effect. Chance fluctuations due to sampling error can also cause variation in population density, but this effect is observable mainly in small populations. And this type of variation is called demographic stochasticity. So demographic stochasticity can cause extinction in small populations is the main worry in small populations, demographic stochasticity. So let's talk about this so you understand what it is. Understand this. Just imagine you flip a coin to decide if an individual dies or not before reproducing. Heads, it survives, tail, it dies. Okay, if there are only 3 individuals in the population, then the chances the population goes extinct is the probability of just getting 3 tails and 3 coin flips, which is 1 in 8. If there are 10 individuals in the population, the chance of getting 10 tails and 10 coin flips is 1 in 1,024. Much smaller, but still not zero. So as you can imagine, the probability of extinction in one generation gets infinitesimally small as the population size increases. So if each generation a population replaces itself according to a large number of stochastic events like coin flips, then random sampling variation in those coin flips will cause random demographic variation in population size. So there's random demographic variation in birth and death rates. Just random variation because there's a lot of coin flips involved. And so in some periods, the birth rate will just by chance be higher than the death rate and the population will increase. And then in other periods, the death rates will be higher than the birth rates and the population will decrease. So they just got this random walk. And it's always possible that that random walk could come down to zero, in which case you have extinction. Much more likely in small endangered populations. So this is a major threat to small populations, demographic stochasticity, this random fluctuation in demographic quantities like birth rates and death rates. So in the presence of demographic stochasticity, in theory, all populations will eventually go extinct due to that stochasticity, but the amount of time it takes can be basically without limit, the larger the population is. Smaller the population, the higher the probability of extinction within any given time interval. So major worry for small populations of endangered species. They can easily go extinct just due to chance, and it is preventable. So even if you calculate the mean trajectory of the population and say, okay, on average, we think, we predict this population should be increasing. But nevertheless, you can have random fluctuations causing extinction, even if the mean predicted trajectory is positive. So the probability of extinction during any particular time period goes down as the population size increases. So you can see that. So if the time interval is 1, well, the probability is pretty low. If the time interval is 10, the probability might be a little higher. The probability of 100, it's higher yet. Time of 1,000, it's even higher yet, because the more time goes by, the greater the chance is that just by pure random events, the population can go extinct. So demographic stochasticity is a major worry for small populations of endangered species. They can go extinct easily just due to chance, and it is completely unpreventable unless you increase the population size. Right. Demographic stochasticity might be the reason bird species disappear at higher rates on small islands than they do on large islands. On large islands, they have larger populations and therefore less variation due to demographic stochasticity, less variation in population size. For example, the Channel Islands of California. If you look at the bird extinctions on large versus small islands, on Santa Barbara, over the last 51 years of bird censusing, 7 of 10 species disappeared. Larger Santa Cruz Island, 6 of 36 disappeared over the same 51 years of censusing. And it's thought that this is just due to demographic stochasticity. Smaller islands have smaller populations, and so they're more vulnerable to random events, random demographic events, random differences in birth rates and death rates. Serious worry also for the Mediterranean monk seal, with population of adults less than about 100 or so most years at the population of Cabo Blanco Peninsula in Spain. The population has shrunken quite a bit to very small areas now left in the Mediterranean area. All right, so apparent demographic stochasticity was documented here in this group of Spanish scientists who are monitoring the Mediterranean monk seal population. You can see there's a lot of fluctuation in the number of counts. So we've got different groups: pups, young, subadults, adult females, adult males, and not known. So you can see all the groups are fluctuating. And, look at this. They did an R graph, and they have the number of counts going down to a negative number here at -10. You can see that these fluctuations are, worryingly close to zero here, with a small population size. So these large fluctuations at some point could, just by chance, come down to zero one year, just due to demographic randomness. So here we have the counts of the number of seals in different groups and the overall trend here in time. And you can see that most of the trend is dominated by decreases, which is mortality. And then in between these long periods of mortality, there's a little bit of reproduction, which just boosts the population up. Okay, so these are numbers that are very close to zero, which is very concerning. All right, so demographic stochasticity puts small populations at immediate risk. Large populations are not at immediate risk, but they eventually could be. But with environmental fluctuations superimposed at the same time as demographic stochasticity, even large populations can be at immediate risk. So this is demographic plus environmental stochasticity operating at the same time, operating in all populations, really. And it's the smaller populations that are at most risk, obviously. So environmental stochasticity, this is just unpredictable environmental changes that could cause density-independent mortality across the entire population, such as storms, unseasonable cold or heat, random changes in current speed or direction that influences larval dispersal or upwelling of nutrients, etc. And these can have large effects independently of population size. Environmental stochasticity can be seen in these phytoplankton I showed you before, the abundance time series, where you have a seasonal effect that's different year to year. So we have both a seasonal stochasticity and then annual stochasticity as well. So this is an example of Chaetoceros and Asterianella diatoms in intertidal sandy beaches, the Pacific Ocean. So environmental stochasticity is experienced by all species, and many have evolved to adjust to it. As, for example, Corey's shearwater, this seabird that responds to environmental stochasticity and sea surface temperature, and primary productivity simply by moving to different foraging regions, showing that behavior evolves to reduce the effects of that stochasticity because that stochasticity otherwise would increase the mortality rate. All right, so you can see that there's a lot of variation from year to year in sea surface temperature in different areas, foraging areas. And from 2007 to 2011, we can see seasonal variation in primary productivity and also a lot of variation in sea surface temperature in different areas, some seasonal and some just annual variation that's difficult to predict. And you can fit these birds with small, very lightweight GPS devices that records their position and other compass bearing and temperature and other data and see where they're moving and relative to depth, relative to sea surface temperature, and relative to primary productivity in different years. And this density is the amount of time the bird spent in each of these areas. So this is the home range here, the dotted lines, I think, and then the foraging areas are here. And you can see they're changing in different years. The depth that they forage of the water where they forage in, and the sea surface temperature of the water that they forage in, and to keep sort of a regular, roughly constant, stay at a roughly constant primary productivity. So this is the home range. The primary productivity at the home range is roughly the same all years, although they're changing quite a bit in depth and sea surface temperature. So they can move in response to changes, variation in the environment, to make sure that they are able to forage to the extent that they need to forage while they're nesting and meet the higher energy requirements during nesting. Right, another cause of variation in population size is immigration and emigration from populations to other populations. So most species can be considered to be a metapopulation. In other words, they exist as a collection of populations that are spatially separated, but nevertheless experience some migration between them. So in other words, most species are not just one large randomly mating population, but rather a collection of populations for which mating is far more likely between individuals within that subpopulation, but nevertheless can sometimes also be between individuals from different populations. So if migration drops close to zero into one population, then that population is free to evolve independently and might undergo speciation or branching of the lineage into a new evolutionary lineage. But in the meantime, there is some migration in both directions. So metapopulation can be visualized with a humorous sort of analogy, like a blinking Christmas tree lights. The light blinks off when the population size goes to zero. For whatever reason, demographic, stochasticity, environmental stochasticity drives the population down to zero. And then it blinks back on again when it's rescued by immigration from another population in the vicinity. So at any given time, some lights are on and other lights are off. But the individual lights that are on or off are always changing. So these populations at different locations sometimes drop to zero, but then a new migrant comes in and rescues them. So if the colonization rate from one population to another is lower than the extinction rate of the population, then not every population gets rescued. So then the number of lights blinking off per unit of time is greater than the number of lights blinking on. And eventually that number on will reach zero and the entire metapopulation will go extinct. So like this. Gradually over time, the number of lights that are on gets smaller and smaller until we have extinction of the entire metapopulation. So in this case, the metapopulation is said to have an extinction debt that it has to pay. In other words, eventually it will go extinct. It might take a very long time, but that's a debt that it has to pay because the colonization rate is too slow to rescue populations that have gone extinct locally. So the recolonization rate might decrease because there's elimination of habitat. So if the habitat is eliminated, then migration might not be far enough to reach the nearest subpopulation. So migrants might not survive to immigrate into subpopulations that need to be rescued. So this is what it would look like. The number of subpopulations that are occupied gradually goes down over time with some random noise around that trend. Some marine examples of metapopulation dynamics. This is here we have the stepping stone model. Migration between subpopulations is unidirectional, like moving along stepping stones in the water in one direction. So surf clam larval transport in this paper by Zhang and colleagues. So these are subpopulations on the Atlantic coast, and the larval transport tends to be from north to south, with a little transport within the population as well. But most transport is from north to south. So populations to the south are easily rescued, but populations up on the north are less easily rescued. So an important consideration in marine populations and marine metapopulations is the current direction. Often larvae are transported passively in the current, and so they're dependent on that current direction for the ability to migrate into suitable habitat. So coral reefs are habitat islands, and each island has got a subpopulation of some species. So coral reefs are a nice example of metapopulation dynamics. And in this example of the Great Barrier Reef in Australia, researchers used a mitochondrial genetic marker to show that populations were genetically different in three regions, north, central, and southern, with different color pattern in the southern region. And that difference was explained by geographic distance separating them. But within each region, populations were not well differentiated. Several populations within each region were not well differentiated genetically, which indicated that migration was more common between reefs within locations, but migration between regions was restricted by distance. They also found that genetic differentiation among younger reefs was greater than among older reefs, indicating probably that their extirpation of reef populations were fairly common, um, with recolonization of individual reefs by genetically different founders. Okay. So during the study, these populations were stable, seemed to be stable, these subpopulations, and there didn't appear to be any extinction debt. But, as coral bleaching occurs and we start to lose reefs, the risk is going to increase. So studies like this are important from understanding and monitoring that process. This shows that as the distance between two fish increases in these different regions, they get more genetically different from each other, indicating genetic divergence, possibly by natural selection. So different fish in different regions are evolving to be a little bit different, slightly different from each other, presumably because of different conditions and resources in different locations. This is incipient speciation going on here, possibly. Another example is sea snakes on reefs in the Great Barrier Reef. So researchers censused snakes at each of 14 reefs, close to every year, found that populations blinked in and out of existence with no discernible pattern. So seasonal effects were not significant in a chi-square test. Neither was protected status of any reef. Some reefs were protected and some were not. So we've got absent and present reefs here. Absent meaning black, and then present meaning showing this hatched pattern. So there's no evidence of extinction debt. The present reefs seem to be, well, within the data that they have, relatively constant. So studies like this help with the design of marine protected area networks. So if you want to choose protected areas such that the populations within the area go extinct, they can be recolonized and rescued by adjacent protected areas, and knowing the colonization rates is necessary for these designs. So you can go to all these reefs and just census the snake population year after year. So tide pools are areas where the tide goes out. There's a flat depression where the water remains. So you have these little pools at low tide, and those are special habitats for species that cannot tolerate exposure. And so there are fish that are adapted to tide pools on rocky shores, and they don't occur anywhere else, just in these tide pools. And for example, tide pool sculpins, one group that are an example of those species. So marine species within one pool are somewhat isolated from other pools. They cannot travel between them outside of the water, but then become connected again at high tide when they're submerged by water. So in this study, the populations of tide pool sculpins were shown to have a slightly higher extinction probability if they had a smaller volume. If the tide pool had a smaller volume, which indicates a role for demographic stochasticity. So a smaller pool volume may indicate a smaller population, and therefore greater susceptibility to demographic stochasticity and random extinction. So we have extinction probabilities here and colonization probability with the tide pool volume. Obviously, there's a lot of noise here. But as the tide pool volume goes up, the extinction probability seems to be going down. I mean, there's a high rate of extinction for small pools and larger pools more intermediate. So they applied these data to a model and showed that as the volume of the tide pool goes up, the probability that the pool is occupied by sculpins goes up. So if the high volume, virtually 100% chance that they'll have sculpins, but lower volume range of intermediate probabilities that sculpins will be there because, okay, lower volume pools have a smaller population, so demographic stochasticity can cause extinction and therefore a higher probability of finding a pool that's not occupied. All right, next subject. We're talking about population interactions. So far, we've looked at the effect of population density on fitness and vice versa within a single population of individuals, all of the same species. Not very realistic because in reality, populations of different species interact with each other, meaning the density of one species population can affect the fitness of individuals in another population of a different species, which then affects the rate of increase in density of that population. So both populations simultaneously interact in this way. So we can imagine fitness influencing the growth rate within each population, which then determines the density. But at the same time, the density of one population may influence the fitness of the average individual in the other population. And we will now consider these mutually interacting systems. So populations of many species are increasing, resources become scarce, and there's a struggle for existence within each species and also between species. Okay, so we've shown the math for this struggle within a species. Now we got to expand it to show two species interacting at the same time and consider the possible outcome of that interaction. All right, so here's our little diagram here showing the interaction between the different levels of organization. So we've got organisms and populations, with fitness being the emergent property of organisms, while we have populations with density, dispersion, and growth rate, and a feedback between the two. So this is just within one population, but we can add a second population of a different species, and both of these are going on at the same time within each of these species. And these two populations are located at a similar place and time, and they are able to interact with each other. So the growth rate and density of one population can influence the fitness of the second population, which influences its own growth rate and density, which then influence the fitness of the second population, which affects its own growth rate and density, which then influences the fitness of the second population. So we've got this feedback going on and constant interaction between the two populations. And for now, we're considering just two populations. Of course, in any real environment, there's going to be not just two, but many more than two, maybe dozens, maybe hundreds of interactions going on like this. But we'll start with two and look at what happens with just two interacting species. So we've got types of interactions. So two species can interact in basically four different ways. The density of one population can influence the fitness of individuals in the other population by increasing it, decreasing it, or by not affecting it at all. So the types of interactions are defined by whether the fitness effects are positive, negative, or neutral. So if one species' fitness increases by the interaction and the other decreases, then one label that's popular for that type of interaction is a consumer resource interaction. So it's a plus minus interaction. Consumers' fitness increases while the fitness of the living resource that they consume decreases. So the predator benefits by this relationship with its prey, but the prey does not benefit by its relationship with the predator. Plus minus interaction. Then we've got the detritivore-detritus interaction. A detritivore is just a species that is a scavenger or consumes dead organic material. And detritus is just dead organic material. All right, so if one species' fitness increases while the other one's is unchanged, then this is a detritivore-detritus interaction. It's also called commensalism interaction. One benefits, the other does not have an effect at all. So the detritivore's fitness increases, or the commensals increases, but the species belonging to the detritus obviously is unaffected because the detritivore eats no living individuals. Okay, so plus zero is one type of interaction. What's a third? Well, if both species' fitness increases as a result of the interaction, then by definition, the interaction is called mutualism. They both benefit from the interaction, both species. And if both species' fitness decreases as a result of the interaction, then by definition, the interaction is called competition. So these are the basic four types of interactions that have been studied in nature. And keep in mind here that the actual functional relationships within these interactions do not define them, rather, only the effect on fitness. So you can call it anything you want, but it's going to be a plus minus interaction in this top row. Doesn't matter what you call it, it's a plus minus interaction. And this is a plus zero interaction, plus plus and minus minus. So these relationships here can be anything as long as the effects on fitness is preserved. So this leads to some counterintuitive results and language, as we will soon see. So let's now focus on the competition interaction and the different ways that a minus-minus interaction can occur. All right, so consumer resource interactions are ubiquitous. They're everywhere. All animals have to eat, but consider consumer resource interactions result in competitive interactions. So if two consumers are dependent on the same resource, then they may be in competition. So increase in the density of one of the consumers may cause a reduction in the resource, which then reduces the fitness of individuals in the other consumer population. Also consider one consumer who consumes two resource populations. What kind of interaction occurs between these two resource populations? Well, if one resource increases, it might increase the fitness and therefore abundance of this consumer. And the more abundant consumer here might reduce the fitness of the other resource as a result. So increasing the fitness of one resource indirectly reduces the fitness of the other resource. So by definition, these two resource populations right here are in competition, even though they don't consume a common resource. So this type of functional relationship between two resource populations being consumed by one consumer population is called apparent competition. So point is, you can get a minus-minus with different functional relationships. This is not the only set of functional relationships that will give you a minus minus. There are others also, we'll see in a minute. All right, considering just the type of competition in which there are two consumers consuming a single resource population, or the classic kind of competition, there are two types of competition usually distinguished in the literature. These are interference competition and exploitative competition. So, interference competition occurs when one competitor interferes physically or chemically with the other competitor to prevent it from consuming a common resource. So, marine examples are many cnidarians, such as anemones and corals, who chemically damage their nearby competitors by releasing toxic nematocysts from their tentacles. So, corals and anemones. You may have been stung while bathing by an anemone. Those stinging cells are used for feeding and also for interference competition against their competitors when other anemones, often other anemones or corals, are nearby. So, studies have shown that some corals are better interference competitors than others, and species can be arranged in order from least aggressive, least dominant, to most aggressive, most dominant. For example, Jamaican hermatypic scleractinian corals. So here's a list of several species here of corals. And if you've got an arrow to the right, that means the coral in the row injures the coral in the column. If you got two arrows like this means that both corals injure each other. And the smaller arrow indicates which species does the most damage. So you can organize these in a sequence of greater to lower interference, competition, dominance. Okay, so if you've got an arrow pointing one direction, that means that the species in this row can injure all of these. Whereas the species in this row can only injure these ones to the right. And there's a few species way down here that can only injure, one or two species up here. On the rocky intertidal, a common form of interference competition is just overgrowing and smothering other members of different species that are in competition for space. For example, the brown alga sea palm. This is a sea palm here in the Pacific. So this is an annual species that produces small sporelings that overgrow on mussels. And when strong waves arrive, they can create a force on the sporelings themselves, which are more tightly attached to the mussels than the mussels are to the rock. So you've got this epoxy glue that's basically gluing them to the mussels that is waterproof, and it basically is impossible to separate them from that glue. Whereas the mussels just have this byssal thread, and it's easy to pull them off the rock. Okay, so these sporelings get pulled off the rock, and they pull the mussels off the rock too, and this maintains open space below the sea palm that allows new sporelings to attach and develop where they are no longer threatened by being crowded out by mussels. So in general, on the rocky intertidal, all attached individuals are in competition for space. Space is a resource because it can be consumed. Whenever empty space is in short supply. So bare rock opens up when waves tear off a chunk of mussels. That space is colonized rapidly by bacterial or an algal film, and larger seaweeds then attach and overgrow on the film. Okay, so that's interference competition. Then barnacles overgrow and kill the seaweeds. Again, interference competition. And mussels overgrow the barnacles and kill them. Interference competition. If sea stars are also present, they can prevent the mussels from coming in at a high enough density to kill the barnacles and other species. So interference competition is ubiquitous and going on continuously among attached individuals on the rocks and occurs usually by overgrowing, but also by poison toxins in some species, such as anemones and corals. Right, exploitative competition is just reduction of the fitness of another species purely by consuming their resource. No interferences involved whatsoever. No physical or chemical interference. Just consuming the resource reduces the fitness of the other competitor. And the resource can be food or space, both of which can be consumed and thereby become less abundant. Okay, and this is done without any physical or chemical interference. So this is ubiquitous in nature also, as many animal species will consume the same resource species. And we can see this in simple food webs, such as the rocky intertidal Pacific food web, in which sea stars and whelks, sea star here and a whelk, this is a snail, a predatory snail, they both consume mussels and barnacles. Okay, and therefore they can deplete these resources and reduce the fitness of the other species. In addition, by chance, the sea star also consumes the whelk itself occasionally. So they're not only competitors, but one is a prey of the other. Right, besides exploitative and interference competition, the category apparent is also used. In this case, there's no common resource being consumed at all. Um, Rather, the resources themselves are in competition through their common consumer. If one resource increases, as we already said, it benefits their common consumer, which is an enemy of both. So if you benefit your enemy, then that enemy will harm the other resource population. So it's a negative interaction. So apparent competition occurs, for example, between two populations that have the same disease organism. If this consumer is a disease organism, and if one population increases, it benefits the disease organism, which then increases and can affect the other population. So this is a negative interaction, and therefore competition by definition. So an example taken from recent research is the sea star wasting disease, which shows sporadic outbreaks in Atlantic and Pacific Oceans. And the most recent outbreak in 2013 has affected the Eastern Pacific, mostly northern, and has affected 40 species of sea stars. So the disease seems to be caused by a kind of virus. Lesions form, followed by loss of body parts and fragmentation and death. So we can probably say different species of these affected sea stars are in apparent competition with each other. So this is a sunflower star and then an ochre sea star, because they're host for the virus that they pass on to each other. So high density of one species in a particular location likely has a negative effect on fitness of another species as a result of passing the virus on to the other species. So this is a negative interaction. So is competition, even though there's not a common resource being consumed by the two or more sea star species. Okay, our next subject, we're going to go into the mathematics of exploitative competition. And mainly we're just going to look at, primarily we're going to look at a graphical visual analysis that is more intuitive. So this theory was worked out independently by two ecologists in the 1920s, two theoreticians, Lotka and Volterra, in the UK and in Italy. So this makes mathematical Darwin's struggle for existence between two species, in which limiting resources become scarce and thereby reduce the fitness of the consumers. But now we consider that limiting resources become scarce because they're consumed by competitors of different species. So we ask, how does this affect fitness? And how does this effect on fitness feed back on population growth in the two species? Does one species get eliminated? Do both get eliminated? Do both coexist? And how can we predict the outcome? Well, we're going to do all of that. So we do this by considering the logistic growth equation, but applying it to both species at the same time. But first, we have to be clear on the idea of a limiting resource. And to see what a limiting resource is, consider a curve of concentration of a resource or abundance of the resource against the fitness of the consumer. Okay, as the resource increases from a starting level of 0, the effect on the consumer's fitness is quite high when you increase the resource. But as the resource becomes abundant, then more increases have progressively smaller effect because the resource is so abundant that the consumer has more than it can use. This is basically usually because there are several resources that a consumer needs in combination, and one resource becomes when one resource becomes highly abundant, it can't be used more unless another resource becomes more abundant. So a limiting resource is a resource that, if it is increased, it increases a consumer's fitness. Okay, as it does down here. So this usually happens at low levels of the resource when it's in short supply, but not at high levels. So at the A region, but not at the B region. So as resources become scarce, they become limiting. So in the struggle for existence, populations increase, resources become scarce, and then they become limiting. Okay, so we start with the logistic growth equation to understand the theoretical conditions in which a competitor can exclude another competitor, and the conditions under which the two competitors can coexist. And we're going to understand this by analysis of population growth. Okay, so we start with the logistic growth equation in the context of consuming a resource. So as time progresses, the population of a consumer increases, first exponentially, then settles down to a maximum size equal to the carrying capacity K. So what causes the size maximum? The size maximum is caused by the consumption rate of the consumer, the consumption rate of the consumer. So its population increases as long as there is resource to consume. When there's no longer a resource that is left over, then the population increase has to stop. So the carrying capacity is the population size at which the consumption rate of the consumer population equals the supply rate of the resource that they're consuming. So the consumption rate equals the supply rate. It's an equilibrium point. Population increases until their consumption rate just matches the supply rate of the resource. Then they can't increase anymore because they need to consume more resource than is coming in by the supply rate. If the population increases anymore, then the consumption rate will be greater than the supply rate, and those individuals will not survive. So mortality then brings the population density back down to where the consumption rate equals the supply rate. So if you get above, then it'll bring back down again. And if the population decreases, then the consumption rate is below the supply rate, then more individuals can be supported by the resource, and the population then increases to where the consumption rate, total of the population, equals the supply rate at the location where the population exists. So now we ask, what happens if we add a competitor? Well, then the entire logistic curve has to move down. So we have consumption of the supply rate. Consumption rate equals the supply rate at the carrying capacity. Okay, above the carrying capacity, the consumption rate is greater than the supply rate. Below the carrying capacity, the consumption rate is less than the supply rate. Okay, if we add a competitor, then the whole curve has to move down. If you add more and more individuals of the competitor population, then the curve for the first species moves down and down more and more. The curve moves down for this first species because the first species sees a lower supply rate of the limiting resource. The supply rate is the same, but a competitor is intercepting some of that supply. So the rate of incoming resource available to the first species is lower. Okay. So there's less resource for the first species to consume, which lowers its fitness. Remember, we're assuming it's limiting, and the result is that the logistic curve is lowered. So the first species also has a lower carrying capacity because fewer individuals can exist at equilibrium because the resource availability is lower, and so the consumption rate must be lower. If enough competitor individuals are added, they can then consume the entire supply of limiting resource all by themselves, and the result is the first species is eliminated. So this is called competitive exclusion. One species excludes the other just by consuming its resource. So if two species are present together and their populations are allowed to grow naturally while consuming the same resource, limiting resource, then we can ask what will eventually happen. Will both coexist or one will exclude the other? And this question was first asked by Gauss, G.F. Gauss in the 1930s, who grew 3 species of paramecium together in all combinations in the lab. Okay, so when the Paramecium caudatum and Paramecium aurelia were cultured together, the aurelia species was excluded competitively. When caudatum and Berseria were cultured together, they both coexisted. Why were these different results observed in these different combinations? Competitive exclusion or coexistence? All right, to answer this question, we consider the logistic growth equation, but applied to 2 species at the same time. Species 1 and 2, each one has its own N and its own K, its carrying capacity. So this will show us how and why one species population will decline to 0 or rise to carrying capacity. So let's start with terminology. We have 2 competitor populations, 1 and 2. Both have a population density N1 and N2. Both have carrying capacities K1, K2. Carrying capacity is reached when the consumption rate of limiting resource equals the supply rate. So there are 2 approaches to solving 2 logistic growth equations simultaneously. One is the formal analytical approach. The other is a graphical approach. So we'll use the intuitive graphical approach first. It's possible to use R to solve these equations and plot the graphical result. If you would like to see that explained, you can consult it in the book PDF on the website, my book on marine ecology. But we're just going to use the graphical approach by hand to solve a system of 2 logistic growth equations, differential equations, and we consider a 2-dimensional plot showing the population densities of both species, N1 on the x-axis and N2 on the y-axis. And we want to know what both population densities are at any given time. So a point on this graph represents the value of n1 and n2 at one point in time. So first we mark on both axes the carrying capacities of both populations in the absence of the other population. So this is the carrying capacity of species 1 right here when species 2 is not present. So it'll increase up to this carrying capacity and stop there. If it's above, then it'll decline to the carrying capacity. If it's below, it'll increase to the carrying capacity. And the same for species 2. So now we ask, what will happen to the carrying capacity of both species as we add individuals from the other species? First, if there's no competition, if neither species affects the other's fitness, then obviously the carrying capacities would be unchanged. If we added the other species, then the carrying capacity is constant and falls on a straight line, horizontal for species 2 and vertical for species 1. So here we have a situation where we have two species that are not in competition. So no matter how much of species 2 there is, species 1 increases to its carrying capacity, same carrying capacity regardless, and the same for species 2. So these are two species that are not in competition. So for example, species 1 might be a blue whale, and species 2 might be a mosquito, and their populations have no effect whatsoever on each other. Then the carrying capacities simply stay constant. So if you start out with values over here, then species 1 is going to decrease down to this level, and species 2 is going to increase up to its carrying capacity. If you're below, then both are going to increase to their respective carrying capacities. But if there is competition, then adding individuals or increasing n of one species will lower the carrying capacity of the other species. So let's start by looking at K2, the carrying capacity of species 2. That's the point on the y-axis where N2 equals K2 right here. Now, if we add individuals of species 1, in other words, if we increase N1, then what happens to the carrying capacity of species 2? It must go down along this line because species 1 is consuming some of the limiting resource of species 2. So there's less resource available to species 2. If there's less resource available, then the number of individuals of species 2 that the resource can support goes down. So adding species 1 reduces the carrying capacity of species 2. So the carrying capacity line goes down, N2 decreases as N1 increases. That line goes down until it reaches the x-axis at some point. Now, any starting point above that line, the starting point represents values of N1 and N2, will result in N2 moving downwards until the line is reached. And any starting point below the line result in N2 moving upwards until that line is reached. So this line is called the zero growth isocline for species 2. If you're on the line, there is no growth, no change in species 2. It's in equilibrium with its resource. So no change is happening, no increase, no decrease is happening along this line. So it's called a zero growth isocline for species 2. So species 2's carrying capacity is a line, not a point, that depends on the number of individuals of species 1, N1, its population density. So just a note, Parenthetically, the concept of carrying capacity now becomes a bit ambiguous. We can now think of two kinds of carrying capacity. K2 is the potential carrying capacity of species 2. It's the maximum possible carrying capacity in the absence of the competitor. But if the competitor is present, then species 2 can no longer reach its potential carrying capacity. However, it still has a maximum population density, and that maximum is on the zero growth isocline. That maximum we can think of as the realized carrying capacity, the carrying capacity that is realized in the presence of the competitor. So this will always be lower than the potential carrying capacity K2. Same argument applies to species 1 in the presence of its competitor, species 2. So zero growth means that at any point on this line, which is joint values of N1 and N2, is a point where the population growth of species 2 is 0. For any point on this line, species 2 is at its carrying capacity and cannot increase anymore. Right, notice where the zero growth isocline of species 2 intersects the x-axis. At this point, N2 has a population density of 0. In other words, its carrying capacity is 0 here. If species 1 can increase its density N1 to this point and beyond, then there can be no individuals of species 2 remaining. In other words, N2 is equal to 0. This intercept on the N1 axis, in other words, the value of N1, where species 2 can no longer increase, I call the shutout level of species 1. The shutout level, that's the level where if species 1 increased to this level, its shutout level, then it has shut out species 2. Shut out means exclude or eliminate in English. If species 1 increases to this value, its shutout level, then it is consuming all of species 2's limiting resource, and species 2 no longer has any limiting resource available to it. So its population cannot increase above 0 if the population density of 1 is at this point or above. All right, so let's now try to predict what a species shutout level should be. We can do this by thinking about its consumption rate of limiting resource. In order to shut out species 2, species 1 must consume the limiting resource of species 2. To shut out species 2, its consumption of that limiting resource would have to be equal to the supply rate of that limiting resource for species 2. If its consumption rate is equal to that supply rate, then there's no longer any supply of that resource for species 2. Then species 2 cannot exist at this location where species 1 is at or above its shutout level. In other words, abundant enough to consume the entire supply rate of species 2's limiting resource. Keep in mind that it is possible for each species to have the same limiting resource or different limiting resources, but if both happen to consume each other's limiting resource, then they by definition are in competition with each other, because as a result of that consumption, they will reduce the fitness of the other. So in deriving the shutout level from consumption rates, we assume that each species affects the other's fitness and therefore population dynamics only through the consumption of the other's limiting resource, which might be the same or different from its own limiting resource. And remember, the carrying capacity, potential or realized, of a species is reached when the entire supply of that species' limiting resource is consumed by either or both species together. Okay, remember this plot we looked at. So let's imagine the limiting resource of species 2 first. Here's species 2's limiting resource. Its supply rate is 50 units per hour. Let's say one individual, species 2, must consume 2 units per hour of that resource in order to live. This means the resource can support a total of K2 equals 50 units per hour divided by 2 units per hour per individual equals 25 individuals of species 2 at potential carrying capacity. Now let's say species 1 consumes species 2's limiting resource at a rate of 1 unit per hour per individual of species 1. What's the shutout level of species 1? Well, it's the number of individuals of species 1 necessary to completely shut out species 2. This is the number of individuals capable of completely consuming the entire supply of species 2's limiting resource. So how many individuals of 1 can consume 50 units per hour of species 2's limiting resource? That would be 50 units per hour divided by 1 unit per hour per individual equals 50 individuals of species 1. So if species 1 reaches a density of N1 equals 50, it completely shuts out species 2. There is no resource left over for species 2 to consume, so no individuals of species 2 can exist. Species 2's realized carrying capacity is 0 in the presence of 50 individuals of species 1. So note that we're assuming that there is no interference competition between the two species. If species 2 is aggressive and kills or prevents consumption of that resource by other species, then it might require a higher N1 in order to be shut out. But we are assuming only exploitative competition. The species affect each other's fitness only through consumption of each other's limiting resource. So all we have to consider are the consumption rates on their respective limiting resources. So now we can calculate the actual shutout level of species 1 based on its consumption rate of species 2's limiting resource. Okay, so the consumption rate of species 1 on species 2's limiting resource is 1 per hour. That's consumption rate of species 1. It's always on the other species' limiting resource. The consumption rate of species 2 on species 2's limiting resource is 2 per hour. The ratio of these two consumption rates is the competition coefficient of species 1 on species 2. This is symbolized by A21. A21 is easy to remember. The first numerical subscript, 2, represents the species whose limiting resource we're talking about. The second subscript, 1, tells us the species that is competing against the first by consuming its resource. So A21, the competition coefficient of species 1 on species 2, is the relative consumption rate of species 2's limiting resource by species 1. Okay. So what are the units of the competition coefficient? They are the units of resource consumed per individual of species 1 divided by the units of resource per individual of species 2, both of species 2's limiting resource. So if we analyze these, the units of resource cancel out in both the numerator and denominator, leaving us with individuals of species 2 divided by species 1. So A21 has the units of individuals of 2 divided by 1. It's like a proportionality constant. It's the number of individual species 2 equal to 1 individual of species 1 in terms of the consumption rate of species two's limiting resource. So if you want to convert any number of individuals of species 1 into an equivalent number of individuals of species 2 in terms of consumption of species two's limiting resource, then you just multiply that number n1 by A21. So now we can find the shutout level of species 1. But first, let's just look at the units here. So the competition coefficient of 1 on 2 is just the ratio of these two, which is consumption per individual of 1 divided by consumption per individual of 2 on species two's resource. Assumptions cancel out, and we got individuals of 2 divided by individuals of 1 relative to number 2's limiting resource. Okay, so if you multiply n1 individuals of 1 times A21, you've got n2 individuals of species 2. So it's just a proportionality constant that converts from individuals of species 1 to individuals of species 2. So how do we find the shutout level of species 1? This is the number of individuals of species 1 that completely shuts out species 2, that is, brings its population density down to 0. What is this number? This is the number of individuals of 1 that is equivalent to the potential carrying capacity of 2. That's namely K2. So K2 individuals of species 2 consume the entire supply of 2's limiting resource. To find the number of individuals of species 1 that achieves this consumption rate, we needed to convert K2 individuals of species 2 to an equivalent number of individual species 1 in terms of consumption of species 2's limiting resource. We do this very simply. The number of species of N1 that satisfies this equation, N1 times A21 is equal to K2. Therefore, N1 is equal to K2 over A21. So this equation is saying that if we consider N1 individuals and the amount of species 2's limiting resource they consume, we multiply that by A21 to find the number of individual species 2 that consume the same amount of that resource. So A21 is just a conversion factor, or equivalently, if we have K2 individuals, then we divide by A21 to get the number of species 1 individuals that consume the same amount of species 2's limiting resource that K2 individuals do. That, by definition, is the shutout level of species 1. That number of individuals consumes the same amount of species 2's resource as K2 individuals of species 2. So the shutout level of species 1 is always K2 divided by A21. And similarly, the shutout level of species 2 is always K1 over A12. So the competition coefficient A12 is just the number of individual species 1 that consume the same amount of species 1's limiting resource as one individual species 2. So the inverse of this, 1 over A12, is the number of individual species 2 They consume the same amount of species 1's limiting resource as one individual of species 1. So in our example, the shutout level of species 1 is K2 over A21, which is 25 over zero point 0.5 equals 50. So that's just another way of writing down mathematically what we're visualizing in this little box here. So a total of 50 individuals of species 1 completely shuts out species 2. Similar species 2 shutout level is K1 over A12, which is 100 over 2, which is equal to 50. Okay, so we can label the shutout levels on the isocline plot and draw the zero growth isoclines for both species. The endpoints for each species are its potential carrying capacity and the other species shutout level. All along this line, population growth for that species is 0. So once we have both zero growth isoclines drawn, we can then analyze the competitive relationship visually and see what the outcome is. We start at any point in the isocline plot. All right, but let's first figure out species 2 shutout level. We go through the same sort of analysis here as we did before. And now we can start any place on the isocline plot and predict what the outcome of competition is. So let's start right here. This point represents joint values of species 1 and 2. From that point, we asked how should N1 and N2 change over time? We draw an arrow in the direction of change depending on which side of the isoclines we're on. So if we're above both isoclines, both N1 and N2 are decreasing. If we're above isocline 1 and below isocline 2, then 1 decreases, but 2 increases. If we're above isocline 2 and below isocline 1, then 2 decreases, but 1 increases. If we're below both isoclines, then both 1 and 2 increase. When we cross an isocline, only one species, 1 or 2, is changing. And if we hit one of the axes, then the other species population density goes to 0. Okay, so starting here, what happens? Both species, species 2 declines and species 1 increases until we reach the equilibrium point, which means that species 1 is shutting out species 2. Okay, if we start here, both species increase until we reach this middle ground, and again, the same thing happens. 1 excludes 2. If we start here, then both decrease until we reach the middle ground, and again, 1 excludes 2. So this is case 1, species 1 wins. In this case, species 1 is capable of increasing to its shutout level, which is here, because that level is below its carrying capacity. If it can increase to its shutout level, then it does and shuts out the other species. Meanwhile, in case 1, species 2's shutout level is above its carrying capacity. So species 2 can increase to its carrying capacity, K2, but no farther. So it never reaches the level at which it shuts out species 1. So the result is species 1 reaches its carrying capacity down here, during which it drives N2 to 0. Okay, so let's switch the isoclines. In this case, the yellow is above and the red is below. Now we switch them so the red is above and the yellow is below. All right, so now what happens? This is case 2. Species 2 wins for exactly the same symmetrical reasons. Its shutout level is below its carrying capacity, K2, while species 1's shutout level is above its carrying capacity. So species 1 cannot win because it cannot increase to its shutout level, whereas 2 can, so it does. Okay, this is case 2, species 2 wins. Case 3, either species can win because in both cases, their shutout levels are below their carrying capacity. So who actually wins in this case depends on the starting point and how fast each species can increase their population density, which in turn depends on their intrinsic rate of increase, R0. The higher R0, the faster the species can increase to its shutout level and shut out the other species. So in this case, the isoclines cross, and that crossing point is an equilibrium, meaning that if you're right at this point, then neither population changes. But if you get bumped off that equilibrium point, then one or the other species will go to its carrying capacity and exclude the other species as it passes the shutout level. So if we start in this region, then species 2 will win. If we start in this region, then species 1 will win. And this is a stable equilibrium. If we get bumped off of it, then we'll tend to return to it. But if we end up on either side, then we have an unstoppable process where one or the other species will exclude the other one. So this is case 3. Either can win because both shutout levels are below their carrying capacities. Okay, here we have case 4. Neither species wins and both coexist. So in this case, both species shutout levels are above their carrying capacities, K1 and K2. And so neither can exclude the other because they only increase to their carrying capacity. They can't increase to the level where they shut out the other species. So they shut out themselves first, you could say. So the two isoclines cross in this case also, but in this case, the crossing point is a stable equilibrium. If you reach that point, and you tend to stay at that point. If you're bumped off it, then the system tends to return back to that point. So if you're here, you'll increase to that point. If you're here, you start anywhere in here, you'll increase to that point. Well, 2 will increase, 1 will decrease to that point. In this case, 1 will increase and 2 will decrease to that point. And if you're out here, then both will decrease to that point and stop. And if you're in here, then both will increase to that point and then stop. So this is a stable equilibrium, and neither species will exclude the other one. So this is case 4, which is coexistence. All right, these 4 rules can be framed in terms of the 2 competition coefficients, A21 and A12. So for simplicity, let's just assume the carrying capacities are the same, K1 is K2. So if A21 is greater than A12, species 1 wins. That's case 1. If A12 is greater than A21, then species 2 wins, which is case 2. If A21 is greater than 1 and A12 is also greater than 1, then both can win, which is case 3. And if A21 is less than 1 and A12 is less than 1, both small, then neither can win, and we have a coexistence, case 4. So generally speaking, if A21 is very large, then that favors species 1. If A12 is very large, then that favors species 2. Of course, the carrying capacities are also important, and the higher the carrying capacity of any species, the more likely that species can win the competition because it allows a high value of N at equilibrium. So if the high value of N is allowed, then that means generally you can increase to a level that will shut out the other species, your competitor, by virtue of the fact that you're consuming that competitor's resource. Okay, so this has been a lot of material, so let's kind of just step back and summarize what we've been talking about. So we're at the population level of organization, and we're focusing on the struggle for existence, which we're making mathematically rigorous and predictive. So a fundamental reason for struggle is competition for resources. So we're making that kind of struggle for existence, namely the competition of resources, mathematically explicit and predictive. So to do so, we use the logistic growth equation, which describes the increase in a population first exponentially, then to a saturating curve that eventually reaches an equilibrium at a constant carrying capacity. And in this equation, the struggle is captured by the constant parameter carrying capacity, represented by K. As the population becomes closer to that carrying capacity, the resources per individual decline, and the per capita rate of increase R also declines. This is the mathematical description of the struggle, Darwin's struggle. So we can express this struggle with two populations interacting in this diagram here, both consuming the other's limiting resource. So this is, as they consume the other's limiting resource, their density affects the fitness of the other species just through consumption of limiting resource. We'll use this diagram for other interactions in a bit, but just focusing on competition, increased density of one competitor reduces the other competitor's limiting resource and therefore reduces their fitness. And this is the interaction called exploitative competition. Both species are exploiting the other's limiting resource, and in so doing, they reduce the other's fitness and rate of population increase. So that gives us this negative interaction, which by definition is competition. And we can describe this interaction graphically with the phase space diagram that was pioneered by Lotka and Volterra. This is the phase space theory. It's just a way of visualizing what's going on using a graph rather than equations. So these two guys, Al and Vito, Al is Lotka and Vito is Volterra, independently figured this out in the 1920s, the same year. They wrote the same paper, basically the same time. So this is called the Lotka-Volterra theory of competition, and they also did a paper on predation. So where the isoclines cross the horizontal and vertical axes determine the outcome of the competitive interaction. Just where they cross the vertical axes determines the outcome. So these points of crossing occur at the shutout level, which is the population density at which one species shuts out or completely excludes the other by virtue of consuming all the limiting resource. Okay, and the potential carrying capacity, the maximum population size in the absence of competitors. So just by looking at the geometry of this situation, there are four cases. In case one, the yellow isocline for species one is above the red isocline, all of it. In this case, species one will win the competition. No matter where you are in the phase space diagram, the two populations will eventually progress to this point right here, which is the stable equilibrium, the carrying capacity of one, where there's zero individuals of species two, so one wins. And happens when the competition coefficient for species one is large and the competition coefficient for species two is small. So species one is the better competitor and it wins. In case two, the red isocline, two's isocline, is above the yellow isocline, which is one's isocline. So in this case, no matter where you are in this diagram, the two populations move to this point right here where there's none of species one left and species two is at its carrying capacity. And this is a stable equilibrium. And this occurs because the competition coefficient of species one is small and the competition coefficient of species two is large. Species two is a better competitor. Case three is when the two isoclines cross, and in both cases, the shutout level is below the carrying capacity. Shutout level here for species two is below its carrying capacity, and the shutout levels for species one is below its carrying capacity. So both species can increase to their shutout levels. So both species can exclude the other depending on the starting conditions. And this occurs when both competition coefficients are large, so either can win. Both are strong competitors, either can win. So it depends on how fast a population can increase and where the starting point is. If the starting point is near the shutout level of two, then two will win. If the starting point is near the shutout level of one, then species one will win. Or if one species is faster, then that species is likely to win. So this is case three, crossing isoclines where both shutout levels are below their carrying capacity. We have an unstable equilibrium here where they cross. It's an equilibrium. Both populations will have zero growth rate at this point, but if they're bumped off that point, then they'll go to one or the other carrying capacities. All right, case four is coexistence. This occurs when both competition coefficients are small, so neither one is a strong competitor. And in this case, the two isoclines cross such that both carrying capacities are below both shutout levels, so neither species can increase to its shutout level and eliminate the other species. It's not possible because they reach their own carrying capacities first. So where the lines cross now, that's a stable equilibrium, and no matter where you are in the phase space, you're going to move towards that stable equilibrium where you have coexistence. So just to summarize, we've got, in terms of the outcomes, we've got these two shutout levels. If the shutout levels is less than carrying capacity for either of the two species, then the species can shut out the other species. If the shutout level is greater than the carrying capacity, then the species cannot shut out the other species. And if both shutout levels are greater than the respective carrying capacity, then neither species can exclude the other, and the two competitors coexist. And if both shutout levels are less than the respective carrying capacity, then both species can shut out the other. And which one wins depends on where the starting position is and which species has the highest reproductive rate. And these conclusions can be interpreted in terms of the competition coefficient. Okay, for simplicity, assume that K1 is K2, both carrying capacities are the same. If one species limits the other species more than it limits itself, in other words, either competition coefficient is greater than 1, then exclusion can occur. This case is 1 and 2. So if both species limits the other more than itself, then we have case 3. And if each species limits itself more than the other species, so both competition coefficients are smaller than 1, then we have coexistence. And basically what this comes down to is that a high consumption rate of the limiting resource reduces availability of the resource for the other species and increases the likelihood of excluding that species. So a high consumption rate means a high value of the competition coefficient. So if species 1 has a strong competitor, then its competition coefficient is high. That's A21. If the second is a strong competitor, then A12 is high. And if we know the values of the competition coefficients relative to the carrying capacities, then we can predict the equilibrium outcome of competition. And remember, this is exploitative competition in which the fitness of both participants is reduced only through the process of consumption of each other's limiting resource. And this resource most often is going to be food, in the case of animals, or nutrients and light, in the case of plants. But it can also be space or refuge areas for predators or dangerous or from dangerous conditions. Any space you occupy that can't be occupied by your competitor. So actually there are 5, not 4 cases revealed by the Lotka-Volterra equations. The last one is a trivial one. Both species isoclines are identical. All right, and this occurs when both species have only one and the exact same limiting resource. In that case, each species carrying capacity is also that species shutout level. Increasing n to the point where all my resources consumed is also increasing n to the point where all my competitors' resources consume. So both carrying capacities are also shutout levels. And this means that both species can reach their respective shutout levels and exclude the other one. So both species are capable of winning the competitive struggle. So that's similar to case 3. So which one will win? Well, the species that wins is simply the species that arrives at its carrying capacity or shutout level first. And which species will that be? Well, that's the species with either a high intrinsic rate of increase, just gets there first, or a low carrying capacity, or some combination. We can't really know unless we actually solve the differential equations. But on the same limiting resource, the species that can increase fastest, high R0, to its carrying capacity, and the lower the K, the quicker it can reach it, that's the species that will win the competition. So this is according to the Lotka-Volterra isocline analysis. All right, that's it for this week. Our topic 8. Next week, we will continue talking about competition and real-world examples of competition and how they're studied in nature, including in the Rocky Intertidal, where some of the classic studies were done. And then we'll continue with additional types of interactions, starting with the consumer resource interaction. We'll also look at the theory relating to that type of interaction. So have a great rest of your day, and we'll see you next time.